Welcome back to my course Industrial Biotechnology. Now today I am going to cover a very interesting topic, that is the cheese production, because you know that nowadays throughout the world cheese has been largely used. Previously it was well spreaded in the western country because most of the people they used to take cheese, but slowly this spreaded in the our country also. First problem is that what do you mean by cheese? Cheese is kind of food product through which we can preserve the milk, protein and fat for longer period of time. Now question come how? Because as you know, the milk, protein and fat, they are very good for human health. But we cannot preserve the milk, protein and fat in the form of milk for longer period of time. Even if you do the pasteurization, then after some time that milk will be spoiled. The cheese is a process through which you can preserve the milk, protein and fat for longer period of time. Because the main reason behind that is that cheese is produced through one process, what you call curd making process. And through this curd making, it produces some lactic acid. And lactic acid, and there is an enzyme called rennet. They precipitate out this milk, protein and fat, and that comes out in the soluble form. And liquid that comes out from this industry, we call it whey. And so this is during this fermentation process, during the curd making process is produced the lactic acid, and as you know, I mentioned before, the lactic acid used as a good preservative. So that is the reason that when you prepare the curd, after that you separate the milk, solid and fat in the form of solids, and then you reduce the moisture content significantly. Then there is a process in the cheese making. I shall show you what you call heating, cooking and also salting process, and through which we can have the different type of cheese. Now you will be very surprised to know that in the world there are more than 500 varieties of cheese available, and some cheese you can use as a spreader, some cheese you can take directly as a food. So let us discuss this process in details. Now cheese is a food derived from milk that is produced by wide range of flavor, texture and forms by coagulation of milk protein casein. It comprises the protein and fat from milk, usually milk of cow, buffalo, goat and sheep milk. You know that here I want to point out that the cheese may be of different color. It may be, if you want perfectly white color, we go for sheep milk. But usually in case of cow milk and buffalo milk, sometimes we add some kind of color also in the cheese making process to give a yellow color. So most of the cheese melt at cooking temperature. The word cheese comes from the Latin caseus, from which the modern word casein is also derived. The earliest source is from the Proto-Indo-European root quath, which means to ferment become sour, because this fermentation during the curd making process is produce the lactic acid. Now what is the classification of the cheese? There are 4 different, broadly it can be classified. One is called hard cheese, and there is soft and semi-soft cheese, and there is fresh cheese, and there is processed cheese. The hard cheese means high acid development and high cooking temperature, because as you increase the cooking temperature, the texture will be more hard, and as you reduce the temperature, the texture will be little bit soft. So it depends, that is why we call it hard cheese. Example are cheddar, Swiss and Romano. These are the very famous cheese, particularly cheddar is largely used in the UK. Soft and semi-soft cheese, acid develops slowly and controls the lactose by washing because if you wash out the lactose, naturally the acid production will be very small, very low, because lactose is converted to lactic acid and minimal cooking temperature. The example are brick, Gonda and feta. This is the example of this soft and semi-soft cheese. Now fresh cheese is the high acid development, as for examples are cottage and quartz. And processed cheese actually high heating temperature to stop ripening, because the one important step of the cheese making process is the ripening. So it usually heat very high temperature to stop the ripening process. Now if you look at how it looks, that you will find here that this is Roquefort, this is feta, this is Gouda, this is mozzarella largely used, then ricotta, cheddar is looks like this, brie, and parmesan. This is a kind of cheese. 6 type of cheese used extremely in the pizza. 6 pizza. This is what you call parmesans. Then we have Monte Tardi, Jacks, then Romano, mozzarella, and provolone and cheddar that use in the cheeses then in 6 pizzas. The ingredients that required for this cheese making process is milk, salt, rennet, and starter culture, because all plays very important role in the cheese making process. Obviously, that milk, quality of the milks, I shall show you how the quality of the milks affect the cheese production process. Now before that, I want to tell you that about the cheese. Cheese is a bacterial ecosystem that is in constant transformation as is matured through the numerous biochemical transformation during the lifespan. So it undergoes a lot of biochemical transformation process during this cheese making process. The milk quality, because milk contains, I told you, the cheese making process is a process through which you can preserve the milk, protein and fat for longer period of time. So milk quality plays very important role, particularly the milk content fats and proteins. Now, if the fats and protein is high, naturally yield of cheese will be high. If milk and protein that is less, then yield will be less. Salting is usually done not only to develop the taste, to reduce the bacterial contamination. Rennet is kind of groups of enzyme, particularly proteolytic enzyme, that is used for separating out the casein, that precipitation of protein. And starter culture, that is largely used, like that is card making process. We use the Lactobacillus casei, Streptococcus laxus, the different type of that organisms we use for the card making process. Now milk from the sheep, goat and cows, marries, and other animal can be used for manufacturing cheese. Each of them contains fat, protein, milk, sugar, lactose, minerals and water. I shall give you a very detailed analysis of this process. And fat is present in emulsion, because we know the fat is insoluble in water. So it should always present in the form of emulsion. But the sugar, minerals and other proteins are usually remain in the soluble form in the milk. The casein in combination with the calcium, this is very interesting. This is typically added during the card making process, that calcium ion is in the form of calcium chloride. And in the normal card making process, you might have seen that when you card the card, it spreaded the water, because as soon as I shall show you the picture. But in case of cheese making process, card making process in cheese production, we add some calcium chloride to make the structure of the card little bit harder. So you can cart it, you can make it in the form of blocks. So 1 pound of cheese would approximately require 10 pounds of milk. This is your product is approximately 1/10. It may be little less or this is more than that, depending on the several factors. Now I can give a typical analysis that that if you how the major distribution of major milk component during the cheese making process. Now suppose we take 100 kg of milk, it contains 3.8 kg usually, that if you consider the cow milk, usually it is, if you consider the buffalo milk, usually the fat content is high. So this is 3.8 kg, that is 3.8% weight by volume. Then or weight by weight, you can say that 3.3 kg of protein and 5 kg of carbohydrate in the form of, mostly in the form of lactose, and 0.3 kg, 0.73 kg of S. S means it is minerals. Now, when we produce, I told you that 10% of the milk that converted into the cheese. Now, if we assume that 100 kg of milk produce 10 kg of cheese, then it contains 3.3 kg of fat, 2.6 kg of protein, 0.2 kg of carbohydrate, and 0.19 kg of S. So this indicates during this cheese making process, most of the fats and proteins, they recovered in the form of cheese, but carbohydrate and as that goes in the mostly in the form of whey. You can see the 90 gram whey contains about 0.5 grams of fat, 0.7 gram of protein, and 4.8 out of 5 the kg of this carbohydrate, and 0.54 kg of S. Now, milk quality plays very important role in the curd making process. That the quality of the milk. Now, question comes: what are the parameters that determine the quality of the milk? The first parameter is the poor flavored milk. If it gives a very bad flavor, that is undesirable. That is not desirable. There is another property called mastitis milk. Or the mastitis milk is that there is a kind of disease that you have in the curd, in the cow, and if the cow is suffering from mastitis disease, then it is quite possible those bacteria may come in the milk. And then milk contain antibiotics, because when cow undergoes some kind of infection, we apply some kind of antibiotics, and these antibiotics is present in the milk that will hinder the bacterial growth. So your curd making process will be affected. And milk contain high bacterial count, that is also undesirable, because if you have a high bacterial count, there is every possibility of contaminations. Then milk contains sediments. Some of the milk, they have the sediment, because that is also not desirable. The mastitis, the explanation is given here. What we call mastitis occurs when white blood cells are released into the mammary glands, usually in response of bacteria invading the in the teat canals. So this bacteria will come in the milk. That is undesirable. So now another important things is that milk contains milk can use in milk a lot of pathogens can grow, and this milk of the pathogens are Mycobacterium tuberculosis, TB, we know that, then Staphylococcus aureus, then Salmonella species, Shigella, and E. coli. These are the different pathogenic organisms that can grow in the milk. So that is the reason why we cannot preserve the milk as it is for longer period of time. So if you reduce the moisture content, then and only then the growth of the organism will be reduced, and at the same time we produce some lactic acid which acts as a preservative, and also it contains some salt, so that this will not allow the other bacteria to grow. Now there is another problem that we have in the curd making process, that is the fudge contamination problem. We know the bacterial fudge that will attack, because this is kind of parasite. This attacks with it will grow on the bacteria and kill the bacteria. And this is if any kind of fudge contamination take place, then it will not allow the bacteria which use for the curd making process to grow. So your curd formation process will be totally affected. Now question comes how you can avoid this kind of contamination problem, or fudge contamination problem. There are several remedies are there. Practice the aseptic techniques. This if your conditions is aseptic, it is possible to avoid the that contamination problem. Rotate the culture daily. So you know that if you use the culture daily, because it is not the same culture you are rotating every day, then it is not it will cause some kind of problem. Using the fuzz resistant media, because if the media has some kind of chemical compound which not allow the fuzz to grow. Direct to fat, we don't use any kind of intermediate system. Directly we add the culture in the vat and fog the room with 200 ppm of chlorine. Let me here tell you that when I talk about the air sterilization process, you can remember that I told that two type of air we have. One is stagnant air, another is moving air. Because the air that is passing through the pipeline, the compressed air, that is we usually pass through the bioreactor for the growth and multiplication of the organisms. Because the organisms can grow only can use only the dissolved oxygen that is present in the fermentation media. But in case of this card making process, this card formation usually take place in the open vat. Now naturally the room in which this card making is taking place, that should be free from the contaminants. So how you do that? We just fog this room with the help of oxidizing agent like chlorine, sulfur dioxide. Like this we can iodine, we can using fogging the room with that, so that no bacteria can present in this room, and so that your culture can grow properly. So this is one app. So another very interesting thing is the mixed culture, because you know that as I was telling about the fudge contamination problem. The fudge is very specific with respect to a particular bacteria. So if you have one bacteria only in your culture, then and if it attacks fudge attacks that bacteria, then the whole process will suffer. Now if you have 2-3 different types of bacteria present in the culture, the even fudge is very specific for one bacteria. The other bacteria will grow, because it will not affect the growth of other bacteria. So usually it is recommended the mixed culture is preferable for the card making process. And remove and dispose of whey every day, because whey is the byproduct of the cheese making process. This is to be removed and disposed of. Then another some kind of calculation I want to show you, that how you can calculate that how much cheese you can produce from the 100 kg of milk. Now it is very interesting, the fat by dry matter ratio, that as it is decreases, then you see that protein fat ratio that will increases, because you know, because dry matter, total dry matter comprises of what? Comprises of fat, comprises of protein, comprises of carbohydrate, comprises of S. So if this ratio decreases, then what will happen? The protein fat ratio will increase. So that ratio will be this. Now yield of cheese means that you know on this yield is always calculated on the basis of 100 kg of milk. The 100 kg of milk, it can produce how much of cheese? How you can calculate? The equation is very simple: percentage of fat plus percentage of protein, and it is multiplied by the P factor. And P factor is totally dependent on moisture content. As the moisture content increases, the P factor value will increases. So here you can see that if the moisture content is 30%, P factor is 1.29. So you have to multiply here 1.29. If it's 31, 1.31, 35% is 1.40. If you keep on increasing, the P factor will increase. Now I have taken a typical example. Suppose one milk contains 3% fat and 2.88% protein, and we want to produce a cheese where the protein by fat ratio should be 1.34. Now question comes that how you can do that. So you have to prepare the milk accordingly. So how you can adjust the fat content in the milk protein, how much you want. We want that, suppose we want 2.88%. So 2.88% is the milk protein. If you divide by F by M ratio, that 1.3, we can easily find out how much fat is required. So that is required 2.14. What is the fat content in the milk is 3%. So how much fat you should remove? 3 minus 2.14, that is 0.86%. So this fat can be easily removed from the milk. You know, there is a process called churning process, and through the churning process, this is how we produce the ghee and other things, because through the churning process we can separate the milk fat, and this fat we can use for making different butter or ghee, like this we can produce. So we can easily through a churning process we can separate out this much of fat from the milk, and then you see that let us assume the moisture content of this cheese is 35%. Then 100 grams of cheese this is 30%. If you look at previous, I can show you the exact calculation of this. This will be 1.40 into, this is we want fat is how much? This is 2.14 and protein content is 2.88. So this is to be added. So if you add 1.40, then this will be, this is 2, this is 0, and this is 5. So if you multiply it by 5, it will get approximately 7 kg. Say approximately 7 kg of cheese. So 100 kg milk is produced about this. This figure is this is little bit wrong. It will come. So this is similarly. This also you can correct. This will be replaced by 2.88 because 2.88 is the protein content. So this will count around 8. So this is, so I want to show you how the moisture content affect the yield of the cheese. If your moisture content is 35%, it is around 7 kg. If it is 65%, it will be around 8 kg. Now, other ingredients present in the cheese making is the rennet, that it is a complex enzyme produced from the stomach of the ruminant mammals, because it is a different type of enzymes and mostly it is proteolytic enzyme. The examples they have given, that is the chymosin. This is a key component. It is a protease enzyme that causes this casein in milk. So that this rennet can be used to separate milk into solid curd for cheese making process. So this facilitated the solidification, because one thing is that when you, what is the purpose of adding culture in this curd making process? I told you it is main purpose is to produce the lactic acid, and since the lactic acid, this acid will reduce the pH. As this reduce the pH, it will precipitate out the protein and fats. But if you use rennet, then further whatever other proteins is there, the further precipitation of proteins also may take place in this system. The rennet is used for the production in most of the cheese. Rennet thrives the temperature 85 to 105 degree Fahrenheit range. It would not deactivate it completely until it reaches 140 degree Fahrenheit. Rennet is available in liquid, tablet and powder form. Now this is how it is available in the market. That is the rennet that I don't like to discuss much in detail. It's written here that it can be taken for, we talk about the animal system, but it also can be from the plant system, fungal system also we can have. And commercially so-called vegetable rennet usually contain rennet from the mold this rhizomucor, the Methiae. Now let us talk about the culture, that what are the organism that is used in the cheese, that curd making process. And if you find that most of the starter today has originated by the lactic acid bacteria, originally present as a part contaminating this microflora. That lactic acid bacteria itself present in the milk, and it is like Leuconostoc mesenteroid, the Leuconostoc lactase, Lactobacillus acidophilus, Lactobacillus casei, Lactococcus species. So different type of things. Now one very interesting thing is that different lactic acid producing bacteria, they develop the different acidity in the reaction mixture. So depending on the type of acidity that you required, whether you required high acidity and low acidity, you can choose your culture. I can give the example that lactobacillus lactis, lactobacillus bulgaricus, lactobacillus lactis, if you use, then acidity develop is about 0.64 to 0.75%. Lactobacillus bulgaricus, it is quite high, 2 to 4%. Lactobacillus acidophilus is 1 to 1.1%. This is A thermophilus, this is 0.7 to 0.8%. The Claisen is agglomerate by acid commencing at pH 5.3 to 5.2. So when pH reaches 5.3 to 5.2, the casein, which is the major protein that present in the milk, that is gets precipitate, and titratability of acidity is greater than 0.52%. So you know that this is the thing. If the acidity increase 0.52%, then the precipitation of the proteins that will take place in the milk. So this is the type of starter: single cell starter, multi-strain starter, mixed strain starter that we use. I already pointed out that we prefer the mixed strain starter to avoid the fast contamination problem. That this is and this is the kind of flow diagram of this process. This is the raw milk. You see that, and then we take it here. We do the pasteurization of the milk. This is very important because pasteurization means it is killing of pathogenic organism. Then we standardize and filtration. Standardization means we add different components here as per the requirement. I told you calcium chloride is very important ingredients in the card making because they make the structure of the card very hard. And then we do the culturing. We add rennet or use the culture here to form the card. And this card, this will be hard, so you can card this. You know that I told you that due to the presence of calcium chloride, the structure is a little bit harder, so you can make in the form of blocks. Then stir it and cooking it. Cooking is done at different temperature. Then you get the liquid. You can separate the liquid, what is called whey, and this you do the milling. Uses the kind of mixing that mixing is there, and then we put some kind of salting here, and then we put it in the different package. So then we do the ripening of the cheese. This is the kind of flow diagram that we have, and this is the different molds. You can see different molds means different structure we can give as per our requirement. The cheese is a fermented milk which a portion of water and lactose has been recovered. 4 steps are there. One is the curdling, draining, pressing, and ripening. So these are the 4 important steps involved in this. So as I pointed out, it depends on the kind of milk used, method of curdling of milk, method of cutting and forming the curd, type of bacteria or mold used for ripening, and amount of salt or seasoning added, and condition of ripening. These are the different factors that affect the cheese making process. Now I already talked about the milk that plays very important role, and the composition and quality of the milk that plays very important role in the. And one thing I forgot to tell you, that I was talking about that in case if you want to reduce the fat content from the milk, you do the churning process. But in case suppose you want to increase the fat content in the milk, then how you do that? So what you do, as I pointed out, the fat is insoluble material. It is not soluble in water. So you have to pass. That fat you pass through the homogenization process so that you can disintegrate the fat particles to the very small sizes so that it can remain in suspension in the milk. This is how we can increase the fat content in the milk. So this is the sterilization we have. We used to do. We can use the raw milk, pasteurized milk, ultra pasteurized milk as per our requirement. We can do that. Use of milk, I already discussed what should be the quality of the milk, and this is how the card making process is taking place here in the open bed. I think in the next part of this lecture, I shall discuss the process of cheese making in details. Thank you very much.